So I'm going to be talking about single dominant epistasis. This is when there is a dominant allele at one locus that masks or modifies the expression of alleles at a second locus. To break that down, this would be the first locus and this would be the second locus. In this case, big A here, since this is an inhibitor, is going to be epistatic to big B or little b. Because if an organism has the dominant allele here, or the inhibitor, then it's not going to be able to progress. It's not going to be able to show either of these two phenotypes. So, in this case, I'm going to be looking at dragons and their wing size. So we have wingless dragons, short-winged dragons, and long-winged dragons. In single dominant epistasis, if you cross two heterozygous individuals, then that will always result in a 12:3:1 ratio of the different phenotypes or different traits. So in this case, we have 12 wingless dragons, 3 long-winged dragons, and 1 short-winged dragon from crossing two heterozygous dragon parents. If we move over here, we see that this genotype looks a little bit different. So in this case, it's going to result in 8 wingless dragons and 8 long-winged dragons. You won't get any short-winged dragons from this cross. So, if you notice, you can see that there are 2 little blanks here, and then a bunch of other little blanks next to all of these letters. That's because if you have the dominant allele here, then it's not going to matter what else is happening here, because the dominant A, for instance, is going to prevent the phenotype from progressing any further. And if you have a dominant B, then in that case, it's going to allow the phenotype to progress further and become a long-winged dragon. So it doesn't really matter what's going on at the second spot.